So today I'm going to talk about data analysis using statistical analyses in Excel. But for now, we're just going to talk about the basics of statistics as we would typically use in a molecular and cellular biology lab. So what are statistics exactly? So statistics is a systematic and a scientific treatment of quantitative measurement so that we can look for significance in our data analysis, in the data that we have collected. There's several stages of a typical statistical investigation. The first and foremost is the way we design our experiment. Our experiment needs to have appropriate negative as well as positive controls and should have enough information about each variable so that we can organize them into appropriate blocks. The entire experiment has to be repeated a minimum of 3 times in order for any statistical analysis to be done. Next, we need to be able to organize our data, which means that we need to understand what independent and dependent variables are present within our experimental design. In general, the more variables are there, the more complex the analyses are going to be. Also, when you add variables to your experimental design, it can cause confounding data, and it can make our analysis more intricate, and it can make it harder for us to decide for significance. Therefore, it is best to organize our data as well as prepare our experimental design in as clean a fashion as possible. Typically, we want us to keep all our variables consistent except for the independent variable that we are going to be looking at. Next, we perform our analysis after the data has been organized appropriately and interpret the results based on that analysis. And finally, when we have enough analyses done and enough experimental data available towards a particular finding, we can present that data in some kind of presentation or poster or paper, whatever it is that we are going to be doing in that particular module. So, the role of statistics in research labs obviously is very vital in understanding the results of our experiments, and it is extremely important in making sure that we are maintaining the integrity of the scientific process. It starts at the beginning when we are designing our research, obviously when we are analyzing our data, and eventually when we are drawing conclusions about our research. So there are 2 major areas of statistics that we would be concerned with in a biological research setting. The first one is descriptive, and they concern development of indices from raw data sets and summarize our collected or classified data based on those indices that we develop. And then we have inferential or sampling statistics, in which case we are going to base our generalization of total population behavior based on the results that we achieve from small groups taken from that population. And this can concern with 2 major issues. One is estimation of population, when we are looking at that, and this is done a lot in evolution and ecology and epidemiology. And then second would be testing of statistical hypotheses, which is what we use it for majority of the times in a biology research lab. Now, when we are collecting data, there are various types of data that can be collected. Some type of data is what we call primary data collection. And this type of data is going to be collected by a person or a lab which has designed the experiment. It could be in the form of questionnaires that are answered by people or participants of that research, or it could be through direct experimentation and investigation. So actual wet lab work that we do inside a lab setting. Secondary data collection is collected by people that have not actually done the work themselves, the actual collection of the primary data themselves. It could be a person, it could be a lab or an organization, and it could be used by that organization, lab, or person for their own purposes. Usually it's done as a beginning process to developing their own research protocols or research design or coming up with their own hypothesis. This is when we do our literature review kind of thing. So this involves looking through newspapers, periodicals, primary literature, actual research articles, that is. We could be looking at a government official's website or the organization's website like CDC or NSF, or looking at any type of published source and then combining all that data that we find about the topic that we are interested in order to know what is already known about it, in order to find the questions that we want to study about that topic, and then eventually using the information that we find through this data collection, we develop our hypothesis that we are going to be testing in our actual direct experimentation and investigation to collect our own primary data. So once we have that data, we want to organize it, obviously. So we want to classify it and organize it. There are, again, several ways that we can organize our data. The first part is basically just arranging our data into sequences and categories or groups based on common characteristics. So, for example, you have data about people's heights from various countries. So you can make a table out of it that shows all that information, and we organize all the data about the height together in one category. And then maybe we have data about a particular disease and its severity in various population, and that would be organized separately in a separate kind of a database or separate Excel sheet, not the same one, in order to separate the two initially so that we can analyze them in the beginning by themselves. You can organize these partially analyzed and synthesized data sequences into tables, graphs, pie charts, scatter plots, or pictures. Any one of those would be a beginning, could be a beginning of organization of data. As we're doing that, we need to think about the variables in each part and categorize them into their appropriate category. There are several types of data that we collect. Some is more qualitative and some is more quantitative. Obviously, the quantitative data is going to be the most accurate in some way, but the qualitative data actually gives us very important and useful information as well. And in some case, that is the only type of data we can get. So here are some categories that you can have your variables be in. One of them is discrete, which is where we are looking at images and we're just basically counting number of cells or projections or the number of protein molecules that are present in a particular sample. So it is the most basic observation. These are kind of like observations that you can make from it. You can have continuous, which would include height, projection length, size of cells. So now you're beginning to get some quantitative measurements. But again, these can be done in a more qualitative or quantitative data. Qualitative data at the end of the day are data sets that cannot be measured on a scale. So they are more just the way things look or the way things behave. So it would be like fragmentation of DNA or blebbing of cells to indicate death. Or, like, for example, presence or absence of a particular morphology thing that we are looking at. Quantitative data, on the other hand, are actual discrete measurements that we can do. For example, changes in weight over time, or measurement of enzyme activity in a particular unit, or the amount of protein or DNA that is present. So some common statistics that we can do once we have these data, and this is going to be again looking at both qualitative and quantitative data, but think about what it is that we can do with each of these, where we can use it. So one of the things we can do is we can measure the central tendency of the group that we are looking at. And we kind of, say, well, what are most of these people doing? How are most of these cells behaving in this particular environment in the presence of UV radiation, when they're zapped by UV, or when we are treating them with a chemo drug, or when we are giving them an antioxidant. And we can do that by measuring the mean or a median or a mode, which is what we call statistical average in some ways, to look at, well, how do the cells, or how do the humans, or how does that organism behave as a whole when given a particular stimulus or under a particular condition. We can also look at measures of dispersion, which goes a little bit further into the data set. And it says, okay, well, on average, the cells shrink in size when they are exposed to the stimulus, but then there is a range of how much they shrink, right? So some shrink a lot, some shrink not at all, others actually would seem to go the other way. So the measure of dispersion looks at the range of the data, how variable the data is within a particular group. So it can be done by looking at the mean deviation or a standard deviation of your data set that you have. Another way to look at that dispersion would be looking at asymmetry or skewness. And that's done by doing distribution analyses. So you can see that most of the cells were shrinking, but then there was a little tail or small population that did not. And then finally, we can look at measure of relationship, which looks at correlation or regression. So here you kind of think about the standard curves we draw, for example, where we are correlating absorbance with amount of protein or amount of a dye. Or you can think about correlation of smoking to, heart disease or cancer, something like that. So I'm going to go a little bit further into each one of these. So for the first one, you have the measure of central tendency. This way, you're going to basically calculate the statistical average. It's not going to reveal the whole story, but it does, because it doesn't show how variable your data is between the two conditions you're looking at, but it is going to give you a little bit of something to look at. So let's say that I have two groups that I'm comparing. And within the two groups, when I compared them, group A and B, I find that the height of individuals in group A is much higher than the height of the group individuals in group B. So this is like the mean height that I'm looking at, right? The mean height would be the arithmetic average of all the values in each one of my groups. So you find that by adding all the values and then dividing by the sum of number of replicates. So if I have 10 people in each group, then I add up all those 10 numbers, right? So I add up all my numbers, sum of those, and divide them by 10, right? And I'll get my average. And then a median would be the value that's in the middle of a series, where when I arrange them from the lowest to the highest. So if I had three numbers, or not three numbers, if I have 7 numbers or something, right? So I have 1, 3, 5, 5, maybe I have 3 5s, then I have 2 6, then my median would be 5, right? 3 numbers below and 3 numbers above it. The mode would be the most frequent number in the series. Again, in this example, it would be 5 because that's coming in 3 times. The other numbers are represented less than that. So yeah, in this case, like I said, you're only going to be looking at a single thing, a single point of interest, and you're just kind of trying to figure out something interesting in that data set to even go after. So that's what you do. And here, for example, like I said, we just do this, and by themselves this tells me nothing, except it seems that the second group has smaller size than the first group. Next, I want to look at I want to go a little bit deeper into the analysis, and I want to look at dispersion. So this is going to start showing me the variability in the data sets. And so it shows out how spread out or variable my set of data is. And that will help me confirm if the results are reproducible or not. So again, let's say that I'm looking at height of individuals in a particular group, or length of cells in a particular group. And the more variable the data I have, the more dispersed it's going to be. In another setting, let's say that I have a number for the amount of activity my enzyme produces under a particular condition, and I do the experiments 4 times, and I look at the numbers, and I then look at the dispersion by looking at the range, right? So I can have the first time and the second time my activity was a value of 3, let's say. The third time it was 3.3, then it was 2.5, and then maybe fourth time it gave a 4. So the average of this would be whatever number, but then that doesn't tell me how variable my data was. When I look at the range is anywhere from 2.5 all the way up to 4. Right? It's the difference between my extreme values. So it's going to be 4 minus 2.5. That's a pretty large range. You can also look at the rate deviation, where you take the average difference of the values in this particular series, and then look at how variable the numbers are from that average. And then standard deviation, which is the one that we use most commonly in our analyses. And this takes into account your sample size in addition to the deviation from the mean. So this is kind of a little bit better because it may be that the first, for one enzyme, I look at the results, I repeat them 5 times. But the second set of results, I only repeat 3 times for a second set of enzyme. And then my standard deviation will take into account both the number of replicates that I have, as well as the actual numbers and how variable they are. So it will give me a better idea of how variable my groups were. The third thing that we can look at is measurement of asymmetry. This again is similar to the deviation in the sense that it starts to look at how variable the data is. And it's going to look at a pattern of outcome for that particular variable. It's going to tell us what values the variable takes and how often. So the typical bell curve that we talk about comes from that majority of the values should be within that, the average and close to it. And then you'll have some strayed, outliers within it. If it is a perfectly symmetric bell curve, that's considered normal and without any skewness or asymmetry. Sometimes our results are positively skewed or negatively skewed, where your mode, median, and mean are more disparate instead of all together, which is what you would see in a normal setting. So if you put the results in a histogram setting like this, and you examine the range at the bottom on the x-axis and how frequently each number was represented in your data set on the y-axis, you get this histogram, which is what you're using for the measure of asymmetry. And normal means no skew. Positive skew means that you had higher, more instances of your numbers in this lower setting. Those were highly frequent, and the other numbers were less represented. Negatively skewed would mean that your higher numbers were more represented and the lower numbers were less represented. Next, we have measure of relationship. So the symmetry that we just looked at, this histogram, which looks at the range and its frequency, can help us figure out the symmetry, obviously, right? That's what's telling us the skewness of our curve. And it can in turn help us understand the relationship between our experimental variables. So correlation is when we see an association between the two variables that we were looking at. And then regression is going to be where we look at cause and effect relationship between the two variables. whether 1 was the cause of the effect that we were looking at, and if yes, to what degree and in what direction, positive or negative correlation. So here is a graphical representation of how that may show up. A positive correlation shows that the higher the first variable, the higher the second response. So the higher the independent variable, the higher the dependent variable's result would be. And then a negative correlation would mean that the higher the independent variable, the lower the response would be, the lower the dependent variable would be. And if there is no correlation, you wouldn't see a pattern emerging one way or the other. So the value of R in this case, which is the way we denote correlation, is going to lie between plus to minus 1. Plus 1 means that they are perfectly correlated, positively minus 1 where it is a perfect negative correlation, and 0 being there's no correlation. Another example for this would be the regression, in which we are looking at the relationship between the two variables, and this is denoted by capital R instead of the lowercase r. If you want to see a regression effect between the two, the value should be close to 1. This is the case when we are looking at standard curves. In a linear regression, the two variables are going to produce a perfectly linear line with a regression value close to 1. In a nonlinear regression, it could be a polynomial or logarithmic equation. You would see a logarithmic curve with the regression value close to 1 and a polynomial equation to go along with it. So it doesn't have to be linear, it could be nonlinear as well. The statistical methods that we are going to be using in our lab, there are 3 different ones that we are going to focus on. The one that we're going to use most often is obviously the student t-test. Then we have one-way analysis of variance, or ANOVA. And then we have two-way ANOVA, which is just another version of ANOVA when we have multiple variables to look at. A student t-test can only be used when you have 2 populations only. So all you have are 2 categories, a single independent variable and a single dependent variable. In this case, as long as you have a minimum of 3 samples per group, you do not need to have the same number of samples per group. So I can have, for my group 1, I can have n equals 3, but group 2 can have n equals 10 for all I care. So that becomes helpful when we are looking at actual organisms or humans in our for our data collection, because sometimes you can't find the same number of people or same number of organisms with the per category. For example, I had an experiment where I was comparing CSF from patients with various grades of astrocytoma or a brain tumor. There's no way that I can get the same number of low-grade brain tumor as I get grade 2 brain tumor or grade 4 brain tumor. They were always going to be different. Usually I had a lot more of the low grade than I had of the highest grade. But as long as I have a minimum of 3 per group, I am good, and I can compare them using the student t-test. The second analysis that we can run is one-way ANOVA. This is done when you have more than 2 groups available. So in my example, if I was just comparing low versus high grade patients, CSF, then I can run a student t-test. Or if I was just looking at normal versus brain tumor and not looking at different types of brain tumor or different grades of brain tumor, then I can just run a student t-test because all I have is 2 groups. That's it. However, let's say that I want to compare normal patients, that is normal brains with no brain tumor, and then I want to compare low versus high grade brain tumor. Well, now I have 3 groups. Student t-test can only be done for pairwise comparison. So I can't do that in here. If I were to do that, I would have normal versus low grade, right? Normal versus high grade. And then I have a low versus high comparison. So I'll have 3 t-tests that I'll have to run. And I would still need to run them, but that's not enough because it still doesn't look into comparison of all the 3 groups at the same time. So to compare the 3 groups at the same time, we run a one-way ANOVA. And we will learn how to do that in the actual class demonstration for this Excel program to run the one-way ANOVA. One-way ANOVA, the only thing it will tell you with its analysis is that whether or not there is something within those 3 groups that is statistically different. So that is indicated by the p-value. The p-value that we want to look for, regardless of whether it's student t-test or one-way ANOVA or two-way ANOVA, is p is less than 0.05. So less than or equal to 0.05. This essentially equates to 5% false discovery rate. So we have 95% chance that the answer we got, the difference we saw, was real, and only 5% chance that it was a false positive. So it will tell you by showing that p-value below 0.05 that there is something that's different about these 3 groups. But it's not going to tell you where that difference is. To know where the difference lies, we'll still need to run the student t-test. And we would do that to see exactly where the difference exists. Another thing that we can look at is two-way ANOVA. Now, in two-way ANOVA, we are going to be looking at 2 different independent variables. So both student t-test and one-way ANOVA look at a single independent variable on a single dependent variable. Two-way ANOVA looks at the effect of multiple groups of 2 independent variables on a single dependent variable and on each other. Example with that would be my population of brain tumor patients. So I have patients with normal brain, low-grade brain tumor, or high-grade brain tumor. And then I have patients that use cell phones, no cell phones, and patients that use cell phones. So cell phone plus and cell phone minus. So these would be patients that do not use cell phone but have normal brains, patients that don't use cell phone but have low-grade brain tumor, and patients that don't use cell phone but have high-grade brain tumor. And this would be cell phones, people who use cell phones but still have normal brain, low-grade tumor, or high-grade tumor. So now I have 2 independent variables, whether or not somebody's using a cell phone without a headset, and whether or not they have a brain tumor. In that case, I can't run a one-way ANOVA because now I have 2 independent variables. So to look for that effect, I'd run a two-way ANOVA instead. In this case, it will compare not only within the groups over here, but also within the groups over here to look at normal brains, CSF of patients from normal brains that don't use cell phone versus the ones that do use cell phone, low grade that don't use cell phone versus low grade that do, same thing for high. And then it will also compare people that don't use cell phone and have normal brains versus people that do use cell phone and have high-grade tumors, for example. So it's also going to be comparing these type of analyses that you wouldn't have seen otherwise. Okay. In this case, however, you must have the same number of replicates in all groups. So many times, like I said, we will have different number of patients for different conditions, for different groups, or different number of replicates for different cell culture conditions. But for one-way ANOVA and student t-test, it doesn't matter. As long as we have a minimum of 3, we are good. We can still run the analysis without having to worry about it. However, for two-way ANOVA, you would then have to use the data set, a subset of the data set, so that you have the same exact number of replicates in each group. So you have to have 3 per, at least a minimum of 3 per group, or whatever it is that you need, and the same number of replicates so that you can run your comparison. Other considerations, so this is again looking at the same stuff that we just talked about. So this, you can use this chart to figure out what type of data analysis you need to perform. Do you have two sample sets that are same or related? This is going into the t-test specifically and looking at how you can decide which type of t-test to run. If you have two sample sets that are same or related, then you can run a paired t-test. An example of that would be sample from the same patient or sample of before treatment and after treatment. Example from cell culture would be the same cells, exact same cells. I test the enzyme activity in a subset of those cells before treatment and then after treatment. So it's the same exact cells. I'm just using aliquot maybe of the serum in there or looking at its activity without destroying the cells before treatment and after treatment. But if they are not the same or related, then you next ask, do you have sample sets of the same size? And yes, if that is the case, I have N of 3 in both cases, there's similar variability, then I can run an equal variance t-test. If I have the two sample sets that have same variances, so their averages and variance shows up the same when we run the f-test, then we can also run equal variance t-test. If we have sample sets that don't have the same n number and or do not have the same variance, then no, we cannot run an equal variance. Instead, we run unequal variance t-test. If you are in a hurry one day and you need to get something done super fast and you are unsure, and definitely it's not a pair t-test, the best course of action would be to run an unequal variance t-test because it would be the more stringent of the two. So what probability value or p-value indicates significance? This is what we had talked about earlier. We want the p-value to be less than or equal to 0.05, which indicates 5% false discovery rate and a 95% confidence that the observation you're looking at is correct and actually significant. So that's all for the Excel basics. We will be practicing this with the Excel case study that will be posted in your assignment.